I think that it's important that we keep in mind that this, whoops, I did the wrong thing. Here we go. That so much of the population is a rare disease. It's a significant amount in that we've really just scratched the surface in terms of developing treatments for many of these diseases. You've seen that the impact that the Orphan Act has had over the past quarter century or so and how important that's been. And I hope we'll have new developments from the FDA and others in the regulatory arena so that we'll have more facilitation of drugs being developed in the area. So why the interest now? Why a growing interest? I think a part of it is obviously that Big Pharma needs to replenish their pipelines, but another important feature is a confluence of that fact with the scientific developments that have been occurring the last decade or so, and more specifically describing more diseases on the basis of their genetic etiology, and specifically more monogenic diseases. Hardly a week goes by without another disease being described on its genetic basis. And what's important from that most of our therapies are not gene therapy yet. We're not attacking the specific genetic mutation, but understanding the biology which flows down from that gene product. And that enables us as drug developers to have targets against which we can develop drugs and be very specific regarding the cause of the disease. If we look at some data from the Tufts Center, that we enjoy a somewhat better approval rate with drugs in this area than in standard or more prevalent diseases where there is not a simple monogenic etiology. I think it could be a lot better, but it does give us an edge that I think is very important for developing drugs in the space. If we look at approval time, it's a little bit better. Whether or not there's priority review for Orphan in green, and then approval time in the States over years has been a consistent difference favoring orphan drugs. Being able to convert earlier clinical trials to pivotal trials is an important feature, and obviously the FDA is crucial with that. And then having the incredible networks that are often initiated by the patient advocacy groups is extremely helpful with clinical trial subject recruitment. And here's a data, and Emil spoke to this in terms of cost. And this $200 million figure for a development cost for a drug is, one, it's dated, but two, it's also for the successes. And when you factor in all the lost research dollars that go into drugs that fail, is when you get the figure that's much greater than $1 billion. But another advantage is that it does cost less to get a drug approved in this space. And I'll talk a little bit more about why that might be. I'm really eager to see what the report from the FDA will show. Tim commented on that. There have been many suggestions over the past year as to what can be done. Accelerated approval, separate review division, being more sanguine regarding the number of subjects to be studied, the reviewers being in the normal review divisions of the FDA or looking at approval packages that could be for a very rare orphan disease in the morning, and then in the afternoon look at a very, very prevalent disease. And so can often lose sight of how difficult it can be to recruit a smaller number of subjects, because as a proportion of the patients with that disease, it still could be a relatively high proportion. More marketing exclusivity and harmonization has been also discussed. The Institute of Medicine last fall came out with a report, and I think this just further added impetus to this area, suggesting that NIH and FDA, as well as other groups, work together. I think biomarkers and surrogate endpoints is a very important feature of this, and Emil touched on it. We often confuse biomarkers with surrogate endpoints, and a surrogate means that it's some measure in the clinical study which is well correlated with clinical outcomes. And we obviously need natural history studies for that for us to establish a marker as a surrogate marker. And we in industry can't do that necessarily all alone. Having NIH and patient groups take the lead with that, I think, is probably the way to go. And that should go a long way to being able to convince FDA that accelerated approval for a particular application is a worthwhile path. And Emil talked about this, that the Fabrazyme has been the only genetic disease where treatment has been approved on the basis of accelerated approval, and this is through kidney biopsies and looking at the lack of a substrate accumulation through that. So a long way to go, but there is precedent. Yep. You want it higher, or? It's not in my way. It's okay? It's not too low? Okay. So let me go to the formation of my unit. I started at Pfizer last June. And so what our mission is to make medicines for patients that have diseases which are both orphan and genetic. For me, having the genetic basis of the disease is key. We all know that drug development is exceptionally risky. And a large cause of that risk is not knowing that your target, that your drug may affect very, very well, is clinically meaningful. And so that you take the development all the way into a phase 2A study, and then you fail because your one drug against one target and one pathway of a disease that may be very, very multifactorial fails to have a clinically meaningful effect. And studying monogenic diseases really turns that completely around, that you understand the disease has this cause, and if you make a drug in that downstream biochemical pathway that's affected by that gene, that mutated gene, and the drug works well against that target, you have a much greater chance of having a meaningful clinical effect. And that the simplest examples are enzyme replacement, that there are many examples in the field right now, where you're replacing a missing protein. Hemophilia is another example where there's a clotting factor that's missing, and you replace it. So we are building a portfolio of medicines across different diseases and different targets and different modalities and development stages. The company as a whole, over the recent year and a half, has had 4 efforts, if you will, that are outlined here that show our renewed commitment, if you will, to orphan and genetic diseases. In the end of 2009, a licensing arrangement for a Gaucher's enzyme with Israeli company Protalix was announced. And so that's a late-stage asset that's in the regulatory process. And then my unit in the middle of last year was formed. And then towards last fall, we acquired FoldRx, and that was acquired for a small molecule chaperone for the polyneuropathy of familial amyloidosis. And that's also in the regulatory process now. That was a phase 3 asset. And the research group from FoldRx was incorporated into my unit. And then a few weeks ago, we announced our first external collaboration with Zaccaron, a small biotech company in San Diego that I'll tell you a little bit about. Commercialized products, Michael showed a slide with, I'm not sure we were on that, but we have several commercialized products in the area, probably best known for factor VIII and factor IX products to treat hemophilia A and B respectively that came through Wyeth. But we also have other products in the oncology area, pulmonary arterial hypertension, growth hormone, acromegaly, MS, and then another oncology drug. There are more examples than this, but this is just a highlight of the more major products. So clearly big pharma, including Pfizer, has a lot of catching up to do, but it's not a new brand new area for a company like Pfizer. The pipeline looks like this. Talyglucerase for Gaucher's disease, enzyme replacement. That's in the regulatory process now, and that's the enzyme through Protalix, and that's handled in a different unit of the company. Tafamidis is the small molecule chaperone for transthyretin, which is misfolded in this genetic disease, and tafamidis restores normal folding so that protein is able to traffic normally. And that's in a separate unit of the company as well. And then within my unit, we have several programs in earlier development. For muscular dystrophy, we have 3 types of approaches. 2 are anabolic in nature. One being myostatin, and against myostatin as the target, we have both an antibody as well as a peptide. And then we have a selective androgen receptor modulator as a small molecule program. And then the DAGR, or dissociated agonist of the glucocorticoid receptor, we're seeing if that has some benefit in some muscle models. For backup pipeline products for the hemophilia products that we have on the market, we have a factor 7A program and a factor 10. This is an important responsibility, if you will, that we feel that to support the programs that we have, the products that we have already on the market, to bring along other drugs to back them up. We have a very active program in cystic fibrosis. This has historically been funded by the Cystic Fibrosis Foundation. This program came to us from FoldRx. It's been gratifying to me in terms of being able to put Pfizer resources behind this program, because when it was at FoldRx was a little biotech company that clearly resource constrained, and we were able to continue the funding from the foundation, but also add some resources and expertise that were not in FoldRx and move the program forward. So it's not yet where it needs to be. My hat is off to Vertex. They've clearly had some very nice clinical data with their compounds, and we hope to develop better compounds that are very mechanistically based for this disease. And then the more recent collaboration that we did with Zaccaron. Zaccaron is a small company with particular expertise in glycobiology, and specifically glucosaminoglycans, which are the substrates that are only able to be partially degraded and accumulate in mucopolysaccharidoses. And what they've done is chosen to attack these diseases not on the basis of replacing the enzyme, the catabolic enzyme that's missing, but affecting the synthesis in a qualitative way so that the remaining enzymes are able to degrade the substrate that is made. The promise of the program is that it's a small molecule, that it would be orally available, and for many of these kids, they have CNS involvement, that the small molecule would penetrate the brain and treat the disease in that organ as well. And then lastly, that because of the biochemistry, which I'm beginning to start to understand, one small molecule can treat 2 or 3 or more of these MPSs. So very exciting, and that's a collaboration where we're not going to try to reproduce the glycobiology expertise that exists within Zaccaron, within Pfizer, but we can clearly bring a lot of other resources that Pfizer has that Zaccaron does not. So we look forward to really developing that going forward.